Yeah, thank you, Ted, for your introduction. I'm very pleased to be here and to tell you about our results that were related to the effect of diet on emotional behavior, especially depression, and our attempts to involve microbiota or analyze the role of microbiota in this connection. Our studies were primarily fueled by epidemiological investigations, two of which are shown on this slide, which showed that there is that bad nutrition, low quality nutrition, can increase the risk of depression. And there's in addition also information of epidemiological nature that obesity increases the incidence of psychiatric disorders, and again including depression. And given the fact that nutrition, diet can influence the composition and activity of the microbiota, it was in a way natural to address the question, does diet-induced alteration of the gut microbiota have an effect on behavioral and brain function changes? Well, this idea was we addressed in the context of the so-called gut-brain axis that has been addressed here several times before, which I think has been known now for some time. Of course, it's afferent neurons, cytokines and gut hormones that represent important information carriers, signaling systems from the gut to the brain, and more recently the microbiota has also changed this network of information carriers, and we now have quite good evidence that microbial metabolites can influence brain function, and we also know that these microbial metabolites can also affect and modulate the other information carriers, especially those derived from the immune system, but also, of course, from the gut endocrine cells. And there's also information that microbial metabolites can act on sensory neurons, especially of the vagus nerve. When we talk about the gut-brain axis, of course, we should not forget this is an information system that works in either direction, and this is quite for some time known that autonomic neurons, neuroendocrine factors, also bring information from the brain to the gut. Well, there is, of course, a number of associative and correlative papers now in the literature that show, and we just heard from Christina in the previous talk, that changes in the gut microbiota have an impact on the brain. And one of the challenges in research in this direction is there a causal involvement of microbial factors in neuropsychiatric disorders? And to analyze such causal links, such causal relationships, we need to resort to experimental preclinical studies, and several possibilities one can use in this context. It's, of course, germ-free rodents and the changes we see in these animals in terms of behavior and brain function that can be used. It's a very elegant approach is to transfer the microbial communities from neuropsychiatric patients to germ-free mice, and of course there are also other possibilities to look into acute changes, experimental induced changes of the gut microbiota, and see what effect they have on brain function and behavior. Well, this is just one slide I have taken from Ted Tinnan's and John Cryan's group, showing that if you transfer the fecal microbiota from psychiatric patients to germ-free mice, you induce also behavioral changes in the recipient animals, and it seems this seems to be a very strong indication that microbial factors have a role on maybe a causal role in behavioral changes and brain functional changes seen in these animals. In our study, which was primarily conducted by Ahmed Hassan, it was coordinated with all our collaborations within the MyNewGut consortium. We studied the effect of high-fat diet on brain function, and especially brain-related behavior. And in these experiments, we fed mice 8 weeks with a high-fat diet with 60% of the energy provided by fat in the form of palm oil. And what, of course, we saw was a change in the microbial community, which you see here. And when we looked more closely into the changes that we saw, we found that the alpha diversity was actually increased. There was, as not unexpected, as it is known to occur in obese patients, the firmicutes to bacteroidetes ratio was increased, and not only were the firmicutes, but also the cyanobacteria increased in the microbial community of high-fat diets. Fed animals, and while lactospiraceae and ruminococcus was underrepresented, which was interesting in the sense that similar changes have also been reported in depressed patients. In a metagenomic perspective, of course, these changes predict particular changes in the activity of gut microbiota, which could have a relationship to signaling to the brain and causing alterations in behavior. I'm not going into detail here, but I would rather like to tell you what we saw and what Ahmed saw in terms of behavioral changes. The high-fat diet-fed mice, of course, increased in weight and in obesity, and the behavioral analysis showed that these mice looked rather lethargic. They moved less in horizontal and in the vertical direction. The high-fat diet reduced the social interaction between the mice significantly, and maybe most important, what we thought is that high-fat diet caused anhedonia as analyzed by the sucrose preference test, so they took less sucrose solution compared to the control fed mice, and the preference was also significantly reduced. And these findings, the anhedonia, the reduced sociability, there was also reduced self-care, and the disturbed locomotion, exploration, and also food intake. There seems to be also a change in the diurnal circadian rhythm of these activities that led us to conclude that high-fat diet mice have a depression-like phenotype. We were, of course, also interested in finding out some changes of a neurochemical and other nature that could explain these behavioral changes, and in collaboration with something Klaus and Julia Mancano at the University of Reading, we analyzed the brain metabolome and saw that factors related to energy metabolism in the brain, but also related to glutamate and GABA, were significantly altered in particular regions of the brain, such as either in the prefrontal cortex or in the striatum, as shown on this slide. We also found that neuropeptide Y expression was changed, especially in the hypothalamus, which was not so much of a surprise, because we know that neuropeptide Y is important in food intake regulation, but it was also decreased in the hippocampus, which could be of more relevance to the behavioral changes we saw. So the findings in this first part of the study can be summarized like as here. We have a change in the microbial community in the terms of the firmicutes increase at the cost of the bacteroidetes. We have changes in behavior that tell us that there's a depression-like phenotype. There's a change in the expression of neuropeptide Y, of GABA, and of other factors that may be of relevance to signaling processes in the brain. The question, of course, is this is also correlative evidence that these factors may have a role, but doesn't tell us anything of a causal relationship. So we really, and Ahmed asked the question, is the gut microbiota really causally or essentially involved in this high-fat diet-induced changes in behavior? And in addressing this question, he chose an approach in which the mice were treated with a cocktail of antibiotics, of 3 antibiotics, which are not absorbed from the gut. So we wanted to make sure that the effect of the antibiotics are confined to the gut and confined to the microbial community in the gastrointestinal tract. But, and of course, analysis of the microbial community showed that there was a very significant reduction of the bacterial load in the gastrointestinal tract. We had shown that before in another study, that this treatment is very effective in reducing the bacterial load. Ahmed then went on to analyze some physiological and behavioral parameters of these antibiotic-treated mice, again fed control or high-fat diet. And what you can see here, and what is important, that the weight gain here was reduced by the antibiotic treatment, as was the obesity. So this was the first finding that we found. And when going on, Ahmed also found that the behavioral changes that were caused by the high-fat diet were much reduced by the antibiotic treatment. You can see this on this slide. This is the time spent grooming, which was reduced. So the self-care that was altered, that was reduced or almost abolished by the antibiotic treatment. And this is the data from the sucrose preference experiment. Sucrose intake and sucrose preference both were significantly reduced by the antibiotic treatment. So this would really tell us that the presence of the microbiota, of a normal composition of the microbiota is required that high-fat diet induces these behavioral changes. We also wanted to know, of course, what could be the factor that antibiotic treatment does these changes, and what could be important. And this, in analyzing this question, Ahmed looked at some of the gut and other hormones, and without going into detail, I'm showing you only this slide, and which, of course, not unexpected, we found that the high-fat diet increased circulating levels of leptin very much, and this was almost completely abolished by the antibiotic treatment, and suggesting that leptin plays an important role, and this was confirmed by experiments in which Ahmed used leptin-deficient mice, the OB mice, and indeed looking at the sucrose preference as an index of anhedonia, which was present after high-fat diet, was very much reduced in the leptin-deficient mice, indicating that leptin apparently plays an important role in these behavioral changes. So this is the summary of the major findings of our study, which I would like to give you. So high-fat diet alters gut microbial community and causes overweight, obesity, and a depression-like behavior. Depression-like phenotype is associated with alterations in the brain metabolome and in neuropeptide Y expression. And the high-fat diet-induced depression requires the presence of the gut microbiota and intact leptin signaling. In closing, I would like to put this finding also in the wider concept of interoception that has been developed the last 20 years or so, and which is basically based on neuroimaging studies in which we know that there is a continuous transmission of multiple signals from internal organs, including, of course, the gastrointestinal tract to the brain. And many of these signals don't reach the level of consciousness. They remain unconscious signals that are important for the autonomic and neuroendocrine regulation of internal organ functions. But from the neuroimaging studies, we also know that these unconscious signals reach the limbic system, reach regions of the brain that are important for emotional and affective processes. And so it seems that signals from internal organs, especially also from the gastrointestinal tract, can in this way have an impact on emotion and maybe also on cognition, as we just heard from the previous talk. So it seems that this concept would in a way underpin neurophysiologically what we call often as gut feelings. And I mean, taking our data into account, it would seem that our brain knows if the gut microbiota changes in relation to alterations in diet. So this brings me to the end of my talk, and by showing my team, I would also especially thank our collaborators within the MyNewGut consortium, Yolanda Sanz and team in Valencia, Sandrine Klaus and team at the University of Reading, and last but not least, Nathalie Delsen and her team here in Brussels. Thank you for listening.
Thank you very much, Peter. We have an opportunity for one or two very brief questions. Please.
I was wondering what constituted your high-fat diet? What were the components of the high-fat diet?
I didn't, but what?
You were just explaining about
what was in the high-fat diet? What were the components of the high-fat diet you used?
The high-fat diet, well, it was one of the slides. I mean, it was the high-fat diet was palm oil, so the fat source. And of course, there was an appropriate reduction in carbohydrate and protein levels.
Thank you very much, Peter.
Okay.